Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today I am going to talk about another important part of type of lipids from the paper Lipid Metabolism. The objective of the present module includes the information about sterile alkyl ethers and sterile alkyl glycosides. We also discuss the presence and significance of sterol sulfates as well as the occurrence and importance of cholesterol glucuronide, the alkyl glycosides and polyglycosides. As far as sterile alkyl ethers are concerned, it was first reported from the sediments of Walvis Bay. These sterile alkyl ethers occurs only in marine sediments, and that too belongs to the Cretaceous age. Sterile alkyl ethers consist of 27 to 29 carbon atoms with either one or two double bonds, and these bonds are ether bound to carbon number 8 and 9 of the alkyl chains. The structures of cholest 5 vinyl 3 beta 3 do decanyl ether has been originated in Pleistocene Atlantic sediments. The occurrence of these type of ethers in the sediments with a high diatom suggests that the unknown diatoms ought to be an unserving biological source. Among the types of the lipids, complex lipids, especially the sterol part, in this module we will learn about the sterile alkyl ethers, the sterile glycosides, as well as cholesterol glucuronide and other sterol sulfates, the glycosides and the polyglycosides. These sterile glycosides are also known as derivatives of glycosylated sterol produced by few bacteria, some animals, most plants and fungi. This family of sterile glycosides includes one carbohydrate unit which is either glucose or xylose or even arabinose allied to the hydroxyl group of solitary sterol molecules. And these solitary sterol molecules include brassicasterol, campesterol, dihydrocytosterol, cytosterol, and stigmasterol. Helicobacter species was exposed to be the principally rich in cholesterol glucosides, approximately 33% of the total lipids, suggesting the importance of these molecules as chemotaxonomic markers. In Acoleplasma xanthum species, which is a prokaryote, presence of cholesterol diglucoside has been reported, suggesting that the sterol glucosides take part in the cellulose biosynthesis. It also exerts as a substrate to generate superior homologs of the cellobiose side type with beta-1,4 linked glucosyl residues, and the ensuing disaccharide is ribbed off and utilized as a primer for auxiliary cellulose elongation. The cholesterol glucuronide has been reported to be isolated from the human liver, and approximately 33 nanomole per gram of wet tissue of cholesterol glucuronide is present in liver. In 1982, Hara et al. Have alienated these cholesterol glucuronide from the acidic fraction of lipids and accentuated that it cannot be distinguished from the ganglioside GM4 by thin layer chromatography. They are apparently orchestrated in the liver, synthesized in the liver, and enters the bloodstream where its concentration is approximately 6 microgram per milliliter. The respite part is being removed into the bile, unserving biological source. The acylated sterile glycosides are formed when a fatty acid is acylated at the primary alcohol group, especially of the hexose sugar, glucose or galactose. It results in the formation of sterile glycosides molecule. 6' palmitoyl beta-D-glucoside of beta-cytosterol is the foremost type detected in the tubers of potato. The concentration is about 51%, whereas 6' linoleol beta-D-glucoside of beta-cytosterol is leading in the extracts of soybean. In these products, other fatty acids were also perceived, and further complex molecules were reported in Pistia stratiotus, which is an aquatic plant, where acetyl group at carbon 2 and carbon 4 acylates cytosterol glycosides adjacent to a sterile C6 residues on the sugar moiety. It has also been revealed that acylated sterile glycosides exist at a concentration between 1 to 125 milligram per 100 gram fresh weight in the vegetable parts, and these parts include the leaf, the stem, the root, tubers, fruits, cereals, and the acylated form is 2 to 10 times more plentiful than the non-acylated sterol glycosides. Cytosterol glucopyranoside acylated with linolenic or linoleic acid from Escherichia chrysantha was demonstrated to boost pesticidal activities, killing Oryzius latipes in 2 hours at 100 ppm concentration. Sulfated steryl glycosides is another important subtype of the complex sterol lipids. These sulfated steryl glycosides is the peculiar characteristics of the starfish. In Lynchia levigata, which is a starfish, various polyhydroxylated and sulfated steroid glycosides have been portrayed. Lincoside, a diglycoside compound, possesses the most unusual chemical structure as xylopyranosyl with lone glycoside moiety and methyl xylopyranosyl as the other unit. The other homologue configuration but with only single glycoside moiety have also been depicted for sulfated steryl glycosides. Sterol sulfates is another important type of complex lipid which are predominant in marine invertebrates. As an estimate, over and above 70 sterol sulfates have been illustrated with compounds bearing 1,3 sulfate groups coupled to the tetracycle core or to the side chain having characteristic antibacterial and antiviral activities. In mammalian cells, the sulfate esters of the cholesterol exist as cholesterol sulfate, and it is found in red blood cells and mostly in keratinized layer of skin, where it plays a role in de-squamation process. Squalamine, which is a compression product of cholesterol 24 sulfate with spermidine in the 3 beta position, has been revealed from the stomach of shark, and it has been noticed that this type of lipid is an efficient antimicrobial agent. It also inhibits the angiogenesis and endothelial cell proliferation, and hence it is valuable for fight against numerous pathologies including cancer and macular degeneration. This molecule, squalene, acts as an immune effector in lower vertebrates, and its presence in WBCs, which is also called as immune cells, makes it pragmatic to contemplate that this molecule squalene evolved in lower vertebrates. In the mitochondria and the nucleus of rat hepatocyte, cholesterol 3 sulfate, a sulfated form of oxysterol, hydroxylated on carbon 25. Afterwards, the sulfated oxysterol has been shown to be an effective switch of lipid metabolism in the hepatocyte of the human. Sterol, mono, di, and trisulfates have also been explained in the invertebrates present in warm waters. From a sponge living in Philippine sea water named Poecilastra laminaris, Annasterol, which is a sterol monosulfate, was sequestered, possessing the antibacterial properties against Bacillus vulgaris. Similarly, from the sponge living in waters of the Bahamas named Petrosia Weinbersterols, also known as sterol disulfates, was isolated, and it has been found that these sterol disulfates or the Weinbersterols possess antiviral properties against the HIV as well as the virus of cat leukemia. Sterol trisulfates are also found in sponges, with the positioning of sulfate groups at carbon 2, 3 and 6. These sterol trisulfates possess remarkable antiviral and antibacterial properties, and a compound from Pseudoaxenisa digitata demospongia revealed anti-HIV activity. Lembesterol, which is another example of sterol trisulfate, was isolated from Petrosia strongylata, which is a marine sponge, and these lembesterols have been demonstrated to possess inhibitory activity against thymidine phosphorylase, an enzyme that is allied to the angiogenesis in solid tumors. These molecules are also used as replica for increasing antiviral properties in the transformed structures. So among the complex type of lipids, there is one subcategory which is glycosides of the fatty acids, alcohols or amino alcohols. In lower organism, for example sponges, in bacteria and in yeast, simple glycolipids are present containing a glycosyl moiety, one or more units allied to the 1-hydroxyl group of a fatty alcohol or phosphoprenols, a hydroxy fatty acid, or to 1-carboxyl group of a fatty acid which is ester linked, or to an amino alcohol. They possess exciting physical or biological assets, and few of them, for example alkyl glycosides, are industrially produced for their detergent properties. Complex glycosides are produced by amide linkage amid a fatty acid and a glycosyl moiety, and hence might be classified into the following key groups. The first group is the O-glycosyl link or ester link, and the second group is the O-glycosyl link or N-acyl link. This subcategory of the complex lipid are poised of a glycosyl moiety, one or more units, which are allied to the hydroxyl group of a fatty alcohol, including either a branched chain or a normal chain or a phenolic alcohol. The glycosyl phosphopolyprenols are implicated in numerous reactions, initiating glycosylation of protein. In this subcategory, there are alkyl glycosides and polyglycosides present. Alkyl glycosides were isolated and deliberated in Anabaena cylindrica, which is a nitrogen fixing cyanobacteria, whereas In the heterosis of cyanobacteria, these alkyl glycosides take part in the protection. Later, some other structures were characterized and studied in additional species, Anamoeba torulosa and Cyanospira ripicae, with different chain length and functional groups. These alkyl glucosides contain sugar moieties glycosidically bound to ketols, ketodiols, and long-chain diols, triols. The A-glycone moiety contains C26 or C28 carbon chains along with the hydroxyl groups at the C3, W1, W3 positions. Ketols and ketodiols enclose their carbonyl functionalities aptly at C3 position. In nostochasy, alkyl glycosides were typified by the presence of C3 ketones and triols. In the eggs and tissues of nematodes, ascarosides have been described. In Parascaris, Equorum, and Ascaris suum, it is formed by a glycosyl moiety linked to a 2-hydroxylated hydrocarbon enclosed between 26 and 33 carbon units. These compounds are supposed to be conscientious for providing chemical resistance to the eggs, as they are plentiful in the internal layer of lipid in the egg shell of the nematodes. It is worth mentioning that ascarilose also exist in the lipopolysaccharide of Yersinia pseudotuberculosis and Pasteurella pseudotuberculosis, involved as pheromones in Caenorhabditis elegans. In Bidens parviflora, which is a Chinese medicinal plant, polyacetylenic glucosides have been described, where the glucose unit is connected to an alkyl chain having 10 to 14 carbon moiety, along with 2 to 3 acetylenic bonds and 1 to 2 hydroxyl groups. They are effective inhibitors of nitric oxide production and histamine release. Cytopyloin, which is a closely resembled compound isolated from Bidens pilosa, have been revealed to avert diabetes in mice via T cell regulation. It has a diol with 13 carbon units and 4 triple bonds. Simplexicides also anode glycolipids isolated from Placartis simplex, which is a Caribbean marine sponge, are poised of a long chain secondary alcohol connected to the disaccharide chain by the hydroxyl group, and these revealed to be non-toxic to the cells and also immunosuppressive by mechanism. Since 1977, these alkyl glucosides are used as a raw material for the synthesis of surfactant. In 1893, it was Fischer who first reported the synthesis of alkyl polyglycosides by retorting anhydrous ethanol and glucose in the presence of hydrochloric acid. The alkyl glucosides are exceedingly efficient surfactant as cleansing agent in washing and are extensively used as auxiliaries in crop fortification formulation and in the cosmetic products. So, in this lecture, we have learned about the different kind of sterile alkyl ethers, the different kinds of sterile glycosides, sterol sulfates, cholesterol glucuronide, alkyl glycosides, and polyglycosides. We have also learned about Annasterol and Weinbersterols. Also, the importance, the occurrence, the nature, both chemical as well as biological, has been described in this module. The importance of these sterile alkyl ethers, the glycosides, the sterol sulfates, as well as the glycosides of alkyl and polyglycosides have been dealt with. In the forthcoming module, we will again continue with the types of lipids and their importance. Thank you.